Hello everybody, my name is AfonsoDuarte and I came from ITQB in Oeiras, Portugal. I work in a laboratory for biological energy transduction there, where I work with structural biology and biophysics, and where we try to understand how molecules work and how molecules interact with each other in the living cell. The Institute, besides this field on molecular level, also have plant science research and animal science research. Today I want to tell you about the role of the grid in structural biology by showing you a couple of case studies, specific case studies that I use currently in the lab. So first I want to show you the importance of molecular modeling applications, how they can play a role, the role of the grid within these applications, and finally I will give you some examples on how we do it. So initially I need to introduce you to the players that we are going to work with in this case study. Those are proteins. Proteins are biomolecules that play a key role in living cells. They are fundamental for the regulation and activation and deactivation of key processes that exist in living cells. And they consist of a simple chain of amino acid residues connected in a linear fashion, like showed here in this square. So you basically have one amino acid number 1 that's connected to number 2, and so on until the end of the amino acids, that you can go from 40 amino acids to thousands of them connected. And these chains, of course, you can imagine like a thread. They will fold and they can form different 3D structures within the cell. And the existence of a structure gives the protein a certain function, and if you have a problem on this structure, disease can appear, or your cells might die just because you have the wrong protein doing the wrong kind of function within the cell. Now in nature you have 20 different kind of amino acids, so each of this box can be represented by a 20 different residues. Now if you imagine the combination, it's a huge amount of proteins you can have, thus a huge amount of folds you can have. So it's very important to understand how can these proteins adopt a certain structure, what is the structure, and how does the structure leads to function. And this is what we are going to talk today. So, like I told you, we have 20 different amino acid residues. They are represented in this square, in this box. So the different ones, and this is just to show you the complexity they can have with different kinds of atoms inside, from carbon, hydrogen, nitrogen, and oxygen. And when they are connected in this kind of chains, and they adopt different folds that can go folds like this one on the bottom of ubiquitin, which is a very small protein, and GFP, which is a little bit larger protein, to huge systems like this one on the right side, and they are all in the same scale, these three proteins. And you can see the variety of sizes and folds that they can adopt. And the complex one, we're here on the right side, it's the one we are working in the lab. It's actually one of the biggest machines that we have within the cell, within the living cell, and it's key, it's fundamental for the respiration chain and cellular level. Now, protein structures, we need to have protein structures. If we want to understand how these machines work and how we can regulate them in case of disease, then we really need to have information in high resolution. High resolution, I mean you get resolution up to 2 to 4 angstroms, so you can look at really detailed level within the protein. And you can achieve this by using two fundamental techniques, experimental techniques. One of them is X-ray crystallography, and the other one is nuclear magnetic resonance, NMR spectroscopy. And both of them can give you information in high resolution in different ways. X-ray will use a crystal, NMR will determine structures in solid, both in solid and in liquid. But they can give you complementary information, and that's a very strong thing that we can use. Now, when you get the structure, and the structures I gave you in the past and before, in the slides before, they show a nice structure, and you have another one here on the top right, so you see that you have a certain fold, certain kind of shape. When you have the file that contains this information, is called a PDB file, so it's a PDB format, and basically what it has inside, it's what we are going to use in the session today. What it has inside is information like in a table. So basically what you have, if you open it's a text file with non-ASCII code, where you have in the first column you have the record type, in this case is atom. This means that in this file we are going to describe each atom of the protein. And for example, in this line what you have is you have atom number 1 in this PDB file, and that's represented by this one. Then we describe which kind of atom, so it can be a nitrogen, it can be a carbon located in the alpha position, and that's just something biochemists will like to know, is what kind of carbon we are looking. It can be located in different parts within the protein, and this is the kind of information you have. You can see later you have carbons that are located in other regions called the beta or the gammas or the deltas and so on. In the fourth column you have the kind of amino acid. This is a tryptophan, for example, and you have this 3 code letter. Then you have the chain name, and that's basically how many proteins you can have within the same PDB. And the number of this amino acid residue within the protein. So this, because this atom is the number 1 in the file, doesn't mean it's the first one in the protein structure itself, because you might just have enough data to calculate from the 11th. Then these 3 columns are the key columns actually, because they represent the coordinates x, y, z. If you have the x, y, z here on this representation, this will indicate where the dot of this atom is located. And if you have all these dots together, you can draw things like this one, like this protein. Then you have more columns that actually represent other molecular properties that we are not going to use today. So I'll just keep it simple and show these 3 fundamental ones. And this is how a PDB format is defined. It's very simple. You can open in any text editor, and this is what we are going to use for the case studies later. So protein dynamics, this is one of the key points when you study proteins. You have the structure like I defined a couple of seconds ago, and now we have protein dynamics. Now the dynamics that the protein has is what defines it to work. So if a protein is static, it just has that structure, it's very hard to work. Like in an engine of a car, you need some movement, otherwise the car will not work. You just get a snapshot of the car stopped. You need it to move, and proteins are like this. So you have dynamics here represented by this movement, for example. It's just a depiction of that was used in the cover of Science some time ago, but this shows how the proteins can move, and this movement can lead to activation. This movement can lead for example, protein-protein interaction. So 2 proteins that are in solution and they should interact with each other, for example, one activates the other, one of them needs to do some dynamical changes to interact. Another key point, very important nowadays, it's protein-drug interaction. So if you administer a drug for a patient, you want this drug to interact, and typically what happens is that this drug is going to interact with the protein in some place, and this will lead to a conformational change, so a change in the dynamics of the protein that leads to a deactivation or activation. And this is a key point nowadays in the study of protein. The other point, and I just focus it, is protein-protein interaction. So this is how proteins communicate between them, and this is a representation here of 2 proteins, 2 very important proteins in human cells. On the left side it's a human HSP90 protein, and the other one in blue it's HA1 protein. It's not really important the names. What's important is that when this protein in here in blue interacts with the protein in green, this protein activates this HSP90 protein to work. So if you actually mutate, if you remove this protein from a cell, HSP90, the protein in green, will never be activated. So it's a fundamental protein to activate others, and as this protein is fundamental for the survival of human cells, both of them are key proteins in human cells, and thus the protein-protein interaction that takes place, it's fundamental. It's very important to understand how they work. And when they interact with each other, you form this so-called binding interface or binding hotspots. And this interaction surface is very important to understand how it works, and we want to know this at a molecular level. And one of the applications that we are going to work today is actually an application that allows us to study protein-protein interactions at a molecular level.